this presentation is to take you on a journey of discovery of science. A considerable amount of this work is original, and what it is concerned with is a doctoral PhD doctoral research which began in the early 1970s and finished with the award of that PhD in 1978. None of this work has been published so far, and one of the reasons that it wasn't published is because at that particular time I was in doubt about the interpretation of some of the pictures which you'll see. So partly because of negligence and partly because of waiting for science to catch up, I have delayed this. Now this whole project started with locomotory disturbances in pigs in Brisbane, Queensland, Australia. As a diagnostic pathologist, I was faced with this problem of trying to work out the etiology of this particular disease. There are many causes of a disease, but the etiology is the primary cause. And it does not necessarily mean that that is the total manifestation of the clinical disease. What it means is that without that primary cause, the disease would not exist at all. Secondary causes and then tertiary causes. They're very important because they can modify the clinical manifestation of the disease. This is of great importance, and I will allude to this during this presentation, which was my PhD project, and then the conceptualization of that only in the last few years into something which really is presented as new paradigm shifts in this disease and other related diseases. This first part that I'm presenting is epidemiology and the notion of causation. After that, I will focus on neuropathogenesis and causation and the interaction of the host with the pathogen. Now, epidemiology and neuropathogenesis are intertwined in many ways. What it began with was a focal source of disease in one farm. By inquiry in the district and the like, it was found that this was a point source. And I will talk to you about the predisposing causes which, in my view, led to the clinical manifestation of this disease. Now, it must be remembered that this is the first isolation of this virus in Australia. Now, this particular disease is caused by a virus, porcine teshovirus 1. This is the first isolation in the early 1970s, and this is the original first publication of that isolation. The isolation of this virus created a dilemma when I went into further research on this, because what I found was a virus which is supposed to be exotic to this country, exotic in the sense that it, the exotic part, the exotic section called Teshin disease, was a disease with fairly high mortality and high morbidity, whereas this disease, which I'll present to you, is of much lesser mortality but still fairly high morbidity rates. But then we get into the dilemma later on when I find out that this disease is widespread throughout the whole of Australia. And that's the epidemiology part of it, where I found that it was endemic. So the question comes, are we faced with two strains of a virus like is present in the current literature? It's present in the current literature and has been for years that there are two strains. What I will be discovering, and you'll discover as we go along, is that there's really only one strain. So that's why I say we've got exotic and endemic situations. So what we're doing here is I'll first do a clinical examination, and then I'll go on to look at the sampling from that, the sampling to attempt to isolate and characterise this virus for the first time in Australia. In speaking about this as an original isolation, it is quite interesting that this has been the only isolation. This virus was first isolated in 1970. It's now 2014, and so there have been 44 years and there's no other report of the isolation of porcine Tesco virus 1 in Australia. During the course of this natural outbreak, I took the brains of the affected animals and I mapped the lesions out both qualitatively and quantitatively using knowledge of neuroanatomy. And so mapping these lesions out, I was able to observe, and viewers will be able to observe, the pattern of disease. Unfortunately, with nervous diseases of man and animals, the pattern of the disease has not been made or done. So this is an original contribution which will form the guidelines for future work on nervous diseases of man, such as Alzheimer's disease and other diseases, in which the etiology, the pathogenesis, treatment, control and prevention still remains completely unknown. This is most unfortunate for Alzheimer's disease because Alzheimer's was discovered in 1907 by Alzheimer, and research is no further advanced than it was in 1907. How many years is that? That's 107 years ago. And the only thing that research can offer us now is things are going to get worse rather than better. As the population ages, the disease is going to increase. Now, a major reason for this stultification or stagnation of research is because of the over-fascination with amyloid. Amyloid is a glycoprotein, beta-pleated proteins. It's a secondary product. It's a secondary cause. Certainly, it does cause serious effects upon the neurons, but it remains a secondary cause. So this over-fascination with amyloid in Alzheimer's disease has caused things to happen like attempts to produce immunoglobulins against amyloid, which is quite ridiculous because the original or the primary etiological agent has never been found. So this is what I mean about causation. This is what I mean about causation. The primary cause, the etiology. And that is why, because the guidelines are not there to be seen, from this topographical mapping, which I'll show you, I am going to present to you qualitative and quantitative topographical mapping of the whole brain using coronal sections, whole transverse sections. This is completely original. It's never been published before, and surprisingly, it should have been, it hasn't been done, and it should have been done 100 years ago. Because if it had been done, it's quite clear that the treatment, control and prevention of Alzheimer's disease would have been solved. Now, before I proceeded with the Watt topographical mapping, I investigated this disease using standard histopathology techniques of hematoxylin and eosin and special stains. Out of that histopathology, I made some new discoveries, and one of them is the Watt cell nodule, which I believe is of fundamental and great importance in the study of neuropathogenesis of viruses and possibly many other agents. After that, I proceeded to the experimental part of the neuropathogenesis, and I began by inoculating the animals through various routes to see how the virus got to where it did, and to see whether it got anywhere else as well. After looking at the various routes, I then went on to a rather large experiment in which I compared virus only with virus plus immunosuppressed pigs. The whole exercise from this rather large experiment of virus versus virus plus cyclophosphamide was to examine the pattern of the disease. Unfortunately, the pattern of the disease has been overlooked for many, many diseases of humans and of animals, but particularly humans, and in the case of Alzheimer's disease. In Alzheimer's disease, the pattern of the disease is completely unknown. In Alzheimer's disease, we don't even know for sure whether it's a transmissible disease. One experiment's been done. In over 100 years, one attempt has been made and published to see whether it's a transmissible disease or not. This is most unfortunate. So, what one is doing with this study between the virus and the virus plus cyclophosphamide is to look at two things: the pattern of the disease, the sequential pattern. It's a study of disease where I looked at it from day 6 onwards and followed the pattern of the disease, virus only and virus with cyclophosphamide. It had two objectives: to study the pattern of the disease, and the other objective was to revisit the two-strain current dogma that's in the literature right now, that there are two strains of Teschen disease virus, the virulent strain and the mild strain. The mild strain is supposed to exist, or suggested it exists here. In other countries they talk about it being there, but they refuse to talk about the virulent strain. Now, what I'll show, by the use of immunosuppressives, that there is only one virus. The virus of porcine tissue virus 1 is a stable virus. It's a genome that's quite stable, and there's very little genetic drift in it. It's generally accepted that this is the case. And so for years, people have been trying to work out how it is that you get a virulent form and a mild form. In the past, some 30 or more years ago, the virulent strain and the mild strain were injected into experimental pigs, and there was no significant difference between them. Now, one could attempt to justify that by saying, oh, the virulent one was weakened by passage. Okay, that's a possibility. But the other possibility is that there is a natural immunosuppressive in those animals at the time that the Teschen virus comes in, and so because of that immunosuppression, there is an elaboration and spread of that virus throughout the central nervous system and peripheral nervous system that otherwise would not exist. It would seem that this virus alone produces rather milder forms like the Talfin form, and under the immunosuppression, under natural immunosuppression, there's a greater elaboration of the clinical manifestations. Presented here are these pigs. There's a small one there, that's larger, and that's about the same size as that one. Now, these are weaner pigs. The disease occurred on a deep litter system in a single piggery in Brisbane. And I will allude to the deep litter system later because I think it was part of the cause. So what I'm pointing out here to you is that these are weaner pigs. That's an early weaner. That is a pig of about 4 weeks, and that's about 8 weeks. So you can see that the disease has a range. It occurs in weaner pigs on a deep litter system in a single piggery in Brisbane, Australia. From that comes a long journey of discovery, which ends by looking at the subcellular organelles of the tissues, the nervous tissues, under the electron microscope in order to discover the real neuropathogenesis of this disease. And from that evolves a new concept about neuropathogenesis, which can be applied to many diseases of the nervous system of both man and animal. Now, before proceeding to the specific neurological clinical examination, I will mention the diagnostic possibilities and the differential diagnosis associated with routine clinical medicine. Now, we're faced here with pigs that are showing locomotory disturbances. Locomotory disturbances can be divided into two main parts. One is neurological disturbances and the other is lameness. Now, I think we will very quickly rule out lameness. So we're left with neurological disturbances. So there are quite a number of etiological agents associated with neurological disturbances. We further divide these neurological disturbances into two parts, infectious and non-infectious. We get a lead into that, solving that mystery, by the clinical examination for body temperature. So if we find that the pigs have a fever, then that will tend to rule in infectious diseases and rule out non-infectious diseases. Now, the non-infectious diseases that come to mind could be arsenic poisoning and a number of other drugs, other chemicals. It could be of congenital origin. Once we've ruled those out, we can then rule in infection. Now, as far as infections are concerned, it could be viruses, it could be bacteria, it could be fungi, could be protozoan, maybe even the famous prion, which is now very popular in the literature, but in many ways misunderstood or not understood. So I will attempt systematically by ruling out and ruling in the various possible diagnoses to reach a pathological diagnosis and an etiological diagnosis. So, as well as pursuing the topographical mapping, I studied the diagnostic histopathology, and by doing that I was able to identify and separate out the various infectious diseases. By studying that, you can get an idea of what the actual etiological agent is, and I'll pursue that later. So the diagnostic histopathology revealed new and original discoveries, which I'll allude to later. So now I'll turn to the clinical examination, the neurological examination of these pigs. These animals have been looked at to see the nature of the locomotory disturbance. They can stand. But when they stand, you'll notice a crossing over of the hind legs, which is an indicator of posterior ataxia or paresis. There's a certain weakness, we call it paresis. You might call it ataxia. So all of these animals showed this evidence when they could stand, but some couldn't stand. Some of them were paralysed, just like the highly virulent Teschen disease. And so one wonders immediately about the two-strain theory, because you've got one virus producing a range of neurological disturbances which one might establish that there's some sort of immune differences between them. So these animals are looked at from a point of view of writing, ability to write, their body to re-establish themselves, and we can see that they can basically do that. They're aware of their surroundings, they're bright and alert, they have a pain reflex which indicates that the circuit through the dorsal root ganglion is intact, and so the spinal reflex is intact. This is testing for pain, and we can see that occurring. We can see that the pig is reacting to it. In fact, it knows we're about to do it, and sometimes even before I've touched it, it reacts. Now this is a hyperesthesia, and this indicates that there are lesions in the spinal cord. Now for hyperesthesia, this certainly indicates that there's spinal cord lesions there. So once again we proceed, we can see that basically the pigs can show various evidence of, see how they can take the weight on their front legs. They're still rather weak on the front legs, but they can take more weight there than before. We will see later in the histopathology that there are actually lesions in this upper part of the spinal cord, as well as lesions down here in the lower part. So it's an overall paresis, hind leg paresis, involving the front legs as well, but to lesser extent. So all of these animals have been given a routine neurological examination of the same nature. Now here we have another pig. The locomotory disturbances are very similar. They're locomotory and it can walk, but it's showing signs here of posterior ataxia, and it's showing evidence of hyperesthesia. You notice how it dips. Pig 5, similar, can take the weight on the front legs, on the back legs, less so. Ataxic. Ataxia means errors in rate, range, direction and force of movement. So these animals are showing evidence of rate, range, force and direction of movement, which is ataxia. So they're all pretty similar, except that some are more severely affected than others, and some are actually paralysed and can't move at all. So this was the range of nervous signs that we saw, and stimulated thought as to the fact that we're dealing with one agent but a range of neurological disturbances, which from logical deduction means that there's a range of immune response. We can see hyperesthesia, weakness and response to the needle just lightly sliding along the top of the body. Now we turn to the experimental transmission of the disease. One of the vital things is to be able to transmit the disease, unlike that which happened in Alzheimer's disease. So here I'm attempting to transmit the disease. You saw the natural outbreak. We grew this virus on tissue culture and we purified it, and we then inoculated it into the brain, and we attempted to reproduce the disease that you saw before in the natural case. This pig here is an early weaner pig. It is showing a bit more severe in the changes. You notice the front legs are crossing over as well as the back legs, which indicates that the lesions are more extensive along the spinal cord. There's some response, pain reflex, but not quite as much. As you can see, it's more severely affected, and it appears as though that reflex is not operating as much as it should. Now there's some hyperesthesia, but in this case, hyperesthesia is not so notable. In the clinical case, that was more severe. One wonders about the level of response with hyperesthesia. Sometimes if the lesions are more severe, maybe there's less hyperesthesia. This pig has got mouth movements. It's more severely affected. It's got mouth movements, and it's got also had nystagmus, and it's got a much more severe manifestation of locomotion. It can walk, but it's showing marked ataxia here. It can walk on its front legs, and so once again it's rather similar to the others, but it's It's a bit more severe in that in the head region here, it was showing mouth movements. And in many of the pigs, they were showing nystagmus. So here's another pig on day 8, which still showing these disturbances, locomotory disturbances. The pigs, again, are bright, alert, and aware of their surroundings. It's taking weight on the front legs. The hind legs are weak, and the tail is not curly. It's a little bit curly there, but not as curly as the curly tail you would expect. So this is at day 8 from this particular pig that was observed for 8 days. These pigs here are part of the differential diagnosis which one alluded to. These are locomotory disturbances. But notice the pigs are very much younger. They're only a few days old, and they're showing a condition known as myoclonia congenita, or shaking pig disease. Now this disease, it's certainly of congenital origin, but it could be inherited. The pathology of this is extensive demyelination. There are some reports that this disease is associated with hog cholera or swine fever virus congenitally, and the other virus which is associated with it is circoviruses. And even though the evidence is disputed by some, it does appear that the viruses could be causing this demyelination. Another locomotory disturbance is found here, where the animal is showing nystagmus. See the way the eyes are moving, apart from the flies? But the mouth cannot be prized open, impossible to prise it open, and there is a muscular spasm of all legs. The tentative diagnosis for this will be tetanus. If these legs relax after the injection of this sodium pentobarbital, which I put in, this would confirm to a large degree a diagnosis of tetanus, another locomotory disturbance. So as you can see, the mouth can be opened up now, and the legs are flexible. So this is a case of tetanus, which is another diagnostic possibility associated with the range of diseases. Here is another natural outbreak in a piglet showing some hyperesthesia. You can see that it's a small pig and probably about 3 or 4 weeks of age. So they're all from this disease, this epidemic. The disease ranged in ages from that early weaner, that 3 or 4 weeks, and they were kept there for 10 to 12 weeks on this deep litter system and showed similar signs. Now obviously it would seem that dose played a role, and then the immune response occurred. The animal is bright, alert, aware of its surroundings, but it's showing these locomotory disturbances. As you can see, when the animal is lifted, there are attempts there, as you can see, the forelegs are taking less weight than some of the other piglets. All of this indicates the distribution, the variation in distribution of the lesions. As you can see, it can walk, it is taking weight, but there's a generalised weakness. It's not a quadriplegia as such, but it's a generalised weakness. The front legs, they can move, as you can see, but once again, there are errors in rate, range, direction and force of movement. This is showing much more severe changes than some of the other pigs. This is a pig which was experimentally used for experimental transmission after tissue culture. At day 10, one can see similar changes as some of the pigs before. Posterior ataxia, which we saw in the natural case. This is a successful transmission of the disease, which is confirming the virus equivalent of Koch's postulates that are used for proving etiology in bacteria. So here's a pig that's showing more generalised effects. You see the way that it's less bright and alert, it's more severe. We now come to another pig which I studied for 44 days. Pig H, I called Harold. It's showing characteristic posterior ataxia at day 1 when it was first examined. It's showing hyperesthesia, marked hyperesthesia, but the tail is curly. At day 23, it's still showing similar signs, but an improvement. At day 44, it's clinically normal. Now, it's fascinating to find out why it recovered. Here is a pig at day 10 that had experimental transmission, and then day 11, the signs were much more severe than at day 10. The animal cannot really stand at all, does for a little while, and then it will collapse. And it can take weight on its front legs, but there is no real control over the hind legs. So once again, it's showing characteristic signs of the natural disease. The inoculum was, the virus was grown on tissue culture, inoculated into the brain, and here at day 11 we can see that it's collapsed. The tail is extended again, there is a pain reflex, and there's hyperesthesia. So we're fulfilling Koch's postulates, the viral part of Koch's postulates. So that's at day 11. At day 12, post-inoculation, one can see that there is an increase in the severity of the nervous signs. The pig cannot stand. And so there is still some partial weight, but the pig cannot control properly either its forelegs or its hind legs, and there's marked hyperesthesia, and once again that tail. The pig is also shivering, and it's showing mouth movements, and it's showing nystagmus, indicating lesions in this part of the central nervous system. This demonstrates the method of transmission of the virus intracerebrally. The animal was anaesthetised using sodium pentobarbital to the surgical stage. And then an incision was made here, and then a drill was used to go through the cranial bone. The injection was then made of about 10 to the 6th TCID virus, and the suture line was then made here. The animals responded okay, and the healing was quite normal. Now we go on to the next figures, still pictures of pig H, which you'll recall began with the pig showing marked posterior ataxia at day 1. At day 23, it was improved, and at day 24, it was normal. This is pig H. So if we go on to the next one, it's showing those changes, which was, there's day 23, beginning, day 23, and then we go to the next one, day 44, and you can see there's no ataxia. The animal is walking normally, and we saw that before in the motion pictures. And now we turn to what's known as the Watt topographical mapping, qualitative and quantitative mapping of the brain, which is quite original, has never been produced in this form in the literature. And one would agree that it provides a vital part of the essential needs when studying neuropathogenesis, and that is to establish the pattern of the disease in the brain. Before this topographical mapping was done, the diagnostic histopathology was carried out, and that will be referred to later. What one saw on histopathology was the perivascular cuffing, represented by a circle, the cell nodule, represented by the dot, and this is represented by hemorrhage, this asterisk. And so one follows the pattern of the disease throughout the whole of the brain by using transverse coronal sections, whole transverse sections, which can fit on a large slide. We will see that there is a pattern of disease in which the lesions are less numerous in the front part of the brain, but more towards the ventral part. All of these lesions were related to the neuroanatomy, and that will be described in the text of the figures. But here I want to show you the actual pattern of the disease, where here we're seeing predominantly perivascular cuffing with associated cell nodules close by. Now, as you can see, the intensity of the lesions is increasing as we go posteriorly towards the midbrain. One can see here, once again, the perivascular cuffing, the cell nodules, and here is a lesion which is called either focal micromalacia, or you could call this lesion a focal microinfarct, and I'll show you the lesion in histopathology later. So these are really infarcts, but they're not caused by embolism. I will show later that they're caused by hypoxia when the virus has destroyed the parent blood capillary endothelial cells. But at this stage, you can see that this focal micromalacia, or what could be called the Watt microinfarct, is closely associated with the perivascular cuffing and the cell nodule, and it's mainly in the ventral part, and it's in the midbrain area, more intense. And look, and you'll see that by and large it's similar on both sides. So when it's similar on both sides, the logical deduction from that is that this particular pathogen arrives there via the bloodstream, and we'll follow that more and more later. So if we go on to the next slide, this figure, one can still see the intensity of the lesions is still strong. There is the microinfarct or micromalacia, focal micromalacia here, and the perivascular cuffing associated with the cell nodule. And once again, it's more towards the midbrain. It's more ventral in the midbrain area, the brain stem area, and less up here in the cerebral hemisphere region. So I am postulating that there's a good reason why these lesions have a predilection for this part of the brain and not the other part, and I'll be referring to that later. Now here in figure 17, about the same density, same quantity of lesions, perhaps a little less than in the previous. Once again, qualitatively, perivascular cuffing, cell nodule, and a bit more distribution, a little bit of some distribution in the cerebral hemisphere, but still mainly confined to this ventral part and the brain of the brain stem. And the hippocampus is obviously affected as well. Here the hippocampus is affected. The changes here are more marked, more severe, in that there's more severe microinfarcts, as I call them, or micromalacia. I call this microinfarct, as I'll refer to later in histopathology, because there's a coagulative necrosis which is related to hypoxia because the blood capillary cells in these cell nodules have been destroyed. So what I will be depicting later, and what I'll find by a combination of light microscope and electron microscope, is that these cell nodules actually represent the presence of the virus within blood capillary cells. And therefore, if you follow the pattern of these dots, you can see the pattern of the distribution of the virus throughout the brain. Once again, you notice that it's rather evenly balanced on both sides, which again tends to provide stronger evidence that this is viremia. It's a blood-borne. And when one goes to the ultrastructure and also to the histopathology, you'll see that it's got a specific fundamental lesion in the cell nodule. This has never been described before. The fact that the cell nodule represents the site of the antigen. Other workers have reported the perivascular cuffing. They've reported the presence of cell nodules, which is characteristic of a virus disease, but they've never looked at why there's perivascular cuffing and what is going on at the blood capillary level. These perivascular cuffs are in small blood vessels, but not blood capillaries. So by logical deduction, one would expect the blood capillaries to be affected as well as the small blood vessels, but they haven't been looked at so far until this publication. So later we will see that here is the micromalacia, which is hypoxia. Infarcts, as you know, are produced by emboli, which block the blood vessels. The evidence here suggests that a similar type of effect is produced, but it's hypoxia associated with multifocal blood capillary endothelial cell necrosis, which produces this coagulative necrosis here, and it's associated with the virus that's present in that cell nodule, which I'll show later. These here are showing evidence of neuronal degeneration. So you have neuronal changes here in this midbrain, much more severe because of the presence, which is indicated by the presence of this coagulative necrosis, which I'll show you later in histopathology, and associated neuronal degeneration. Now, this will be investigated further to produce new discoveries associated with this neuronal change. That neuronal change is associated with the hypoxia and not, I repeat, NOT, not with primary invasion of the virus into the neuron. Now, the primary invasion of the neuron is the current dogma in both Teschen-Talfen disease in the current literature, and it's also the current dogma for human poliomyelitis. And I will show that, in fact, this neuronal change is produced by hypoxia because the parent blood capillaries are producing hypoxia to these dependent neurons. And this is also reflected in the coagulative necrosis, which is characteristic of a hypoxia, not of a viral invasion. A viral invasion is characterized in this virus by lysis, liquefactive necrosis, not coagulative necrosis or a partial degeneration of the neuron, which they're showing various degrees of degeneration of the neuron, but not lysis. That lysis, the liquefactive lysis, is demonstrated in the parent blood capillary cells within those cell nodules. And we'll demonstrate that in due course with histopathology, then ultrastructural studies. But here it shows a great intensity of the lesions in that area. And later I will attempt to provide scientific evidence for why this is occurring in this particular spot. In other words, nominating the predilection sites and why they're occurring. And one will mention that this appears to be a physicochemical effect of changing the blood flow as opposed to specific chemical receptors, which is the current dogma. With chemical receptors, the flaw in that argument is that the chemical receptors, specific receptors, can be found where the disease is not present. And if that's the case, then the case is flawed. Whereas with this situation, there is a correlation between the capillary necrosis and the associated degeneration of neurons, and also demyelination, which is a characteristic lesion of this disease, which hasn't been given enough attention so far. And the lesions that are occurring in the dorsal and ventral roots, in the dorsal root ganglia, and also in the spinal cord. There is subtle but specific demyelination, which is a characteristic of this disease. Now, when PIG-H recovers, there are various reasons why this pig recovers. And one of the reasons proposed is that there's remyelination of those demyelinated areas to permit that animal to walk normally again. So this has great ramifications in human disease, because one can see that it is possible for an animal model to recover, and it's also possible for a human patient to recover. It's of great significance clinically. And there are other reasons why Pig H, Harold, recovered, and I'll go into that later. So we now proceed with the topographical mapping, the qualitative and quantitative topographical mapping, which shows the pattern of the disease, which is absolutely vital when one is studying neuropathogenesis, particularly studying the mysteries of the etiology and general causation. So here we can see that most of the lesions are in this area, and these areas are rather devoid, except in the hippocampus here, devoid of lesions. The question is, why is this happening? I'll proceed later, and you'll see that as we proceed quarterly, we're in the vital areas of the brain concerned with basic metabolism, high energy areas, therefore high energy requirements, therefore high energy blood requirements, therefore high needs for oxygen. And so what happens in the body is that there's a variation in the complexities of the blood capillary cells and the networks. There's an increase in the blood capillary networks in vital areas, high energy areas. That's only would seem to be natural, that should happen. And in fact, it's been shown that that's the case. And the surface area in these particular areas of the brainstem have a much higher, greater surface area of the blood capillary cells than in other areas. Because of the increase in surface area, this makes them more vulnerable to friction and slows things up. And I'll show you later that the cells, the leukocytes, and even the thrombocytes, the blood cells, develop villi or pseudopodia, and also that occurs in the blood capillary endothelial cells. And so you have these villi with increased surface areas which are added to the high surface area of the blood capillaries. And so there's a general slowing down of the blood and a greater random chance, in my view, a greater random chance for the virus particles to get off their ride that they've had on the leukocytes, which I think they have, and hit the certain blood capillary cells. So here we see the neuronal degeneration associated with the cell nodule, which reveals is present the virus in the blood capillary cells, and there is the perivascular cuffing. Here in slide 20, one sees another lesion, which I've called the microinfarct of Watt. And the reason for calling it that is to draw attention to this new type of lesion and the new concept of its pathogenesis. Later on in the histopathology, we'll see that this is actually coagulation necrosis, and coagulation necrosis is by definition associated with hypoxia, not with virus invasion. So this once again consolidates or reinforces the concept that this disease begins in the blood capillary endothelial cells and not in the neurons or elsewhere. Once again, one can see the close association. A more severe form here, a lot more blood capillaries affected, and perivascular cuffing. Later on, I will talk again about the evolution of what I've called the Watt theory of relative blood capillary circulation pathology, in which this can be defined as the rate of morbidity and mortality in the dependent cells, that's the neurons and glial cells, nerve fibres and the like, varies directly with the severity of the mortality in the parent cells, that is, the blood capillary endothelial cells that supply them. And so this is an explanation for severity of changes. What I'm proposing here is that there's a greater number of blood capillary endothelial cells showing necrosis, showing death, and that's reflecting in the hypoxia of these areas, much more severe here. And so not only can we, by studying these patterns of diseases, we can conceptualise the severity and explain the severity of the lesions as well as the distribution of them. Now we're moving from the midbrain, where there are quite a large number of lesions, to the medulla oblongata, another vital area, as you well know, and therefore likely to be, and if my theory is correct, then good reasons for finding many lesions, because these are vital areas with a high complexity of blood capillary cells which are needed to provide the energy for basal metabolism. So here we have high numbers with considerable neuronal degeneration in that area, near the cerebellar peduncles, considerable perivascular cuffing, and cell nodules. It must be remembered that when one sees perivascular cuffing, one is not seeing the fundamental lesion, because there's the Virchow-Robin space in there which prevents the virus from moving over into the rest of the neuropil, whereas with the blood capillary endothelial cell, as you know, it's adjacent. There's only the cytoplasmic membrane for the virus to get in. So that's the reason, it's anatomical. That's the reason why it's more likely that the virus will get into the blood capillary cell rather in the small blood vessels associated with perivascular cuffing. But perivascular cuffing is an indicator, and almost invariably with viruses one sees cell nodules. Unfortunately, research workers in the past have not really looked at that cell nodule. They've just noted that it is a cell nodule or a glial nodule, and they haven't examined it closely enough to reveal the mysteries of Mother Nature. The mysteries of Mother Nature are that virus is showing a initial lesion in the body, in that cell nodule, and therefore that's a vital part of pathogenesis, neuropathogenesis, because the definition of neuropathogenesis is the initiation, the mechanisms underlying the initiation of this virus in the body and its subsequent spread. That's what, in case of neuropathogenesis, obviously in the nervous system, pathogenesis in general, the same condition, same explanation. Here we have considerable neuronal change, perivascular cuffing, cell nodules. The same here. And roughly speaking, a fair balance between, not so much there in that case. Quite often it depends upon where you actually slice it. It might be just out a little bit in the way that it's actually sliced. But in general, there's a balance between both sides as you go down the brain, which is indicative of a viremia. That was confirmed in my research, finding of the virus in the blood capillary endothelial cells, and that plus the histopathology, this plus the histopathology led me to go looking in the cell nodule to find what I hoped would be there, and I found it. So here we're in the medulla oblongata, and there's considerable neuronal degeneration and some perivascular cuffing, but it's quite severe in these areas here. This is the cerebellum. One can see that the lesions here are not very high in number. In some other virus diseases and Teshand, severe form, other workers talk about more severe lesions in the cerebellum, but the evidence here does not support that the cerebellum is severely affected. Now we come to the spinal cord. Sections were taken from the more cranial parts, the mid part and the caudal parts of the spinal cords, and lesions were found in all parts right down the spinal cord. The explanation for the clinical signs is somewhat controversial in a sense, because lesions are present. The clinical signs show that it's mainly a posterior paresis or paralysis, but in some cases it may be more severe in the forelegs. But mostly, as you saw in the clinical cases, the animal could take its weight on the forelegs, the front legs, I mean, F-O-R-E. And so it's really, again, a matter of severity of those lesions, the number of blood capillaries that are affected. And it's quite possible that the blood capillary network might be greater in the hind legs than in the more towards the front, because of greater energy needs for them. Certainly in man, walking as he does on two legs, one would think that there'd be a higher demand, high energy demand, where the legs are in relation to the muscular nervous supply in hind legs. But at the moment this is in need of further research. So again, one sees the characteristic lesions, perivascular cuffing, the cell nodule, and the neuronal degeneration. As I mentioned at the beginning, when we were talking about the clinical medicine, we were dividing, as far as diagnosis is concerned, we were dividing or classifying the disease into several parts to try to reach the etiology. And these pigs were characterised by the presence of fever, and I said that because of that, it was more likely that it was an infectious condition. It could be bacteria, virus, fungi, protozoan, or the famous prion. So when one looks at diagnostic histopathology, what one sees here is a characteristic non-purulent inflammation. A non-purulent inflammation means that there are mononuclear cells there, mainly lymphocytes, sometimes plasma cells at later stages when the antibody is being formed. So here is a non-parallel information with perivascular cuffing, and here is early neuronal degeneration with central chromatolysis. So this, we've now narrowed the etiological diagnosis by a process of elimination down to a virus. And so that allows us to proceed with confidence when we take samples to try to isolate that virus from the various parts of the brain that we take. Brain, spinal cord, and peripheral nervous system. Here is a more advanced stage of neuronal degeneration, and you can see these vacuolar type arrangements there. My interpretation of that is the presence of water, that this neuron has undergone degeneration, is suffering from dysfunctional transport problems, pathology, which is derived from the blood capillary degeneration, lack of oxygen and other nutrients. So here is dysfunctional transport system of the neuron. This here is much more severe, obviously, than that one there. And so you've got ranges of variation in severity, and I explained them before in the sense of the numbers of blood capillary endothelial cells of the parent cells related to the dependent cells, these cells here. Again, central chromatolysis, high power, and chromatolysis here, with some increasing gliosis there, scattered but not huge. Now here we see once again this diffuse chromatolysis of the neuron, early neuronal degeneration. Here we see involvement of the myelin. Scattered throughout the neuropil are these odd cells, just a few cells, known as myelinophages, and they indicate that there is myelin degeneration. So this disease is definitely associated not only with neuronal change, neuronal degeneration, but with myelin degeneration, and of course that affects the impulses, the normal impulses, and so that could account for the ataxia that's seen because of the lack of myelin. So these are rather subtle lesions, but they're very important to know about, and quite commonly research workers are not aware of their presence. So myelinophages. This is the cell nodule, which in recent years I've named as the cell nodule of Watt, or the Watt cell nodule, to bring attention to the great importance of this lesion in neuropathogenesis. Quite often this cell nodule is just noted by research workers, and its true significance is overlooked. The research workers quite often go looking elsewhere in the neurons, for example, to try to identify the virus or other pathogens, and they might use fluorescent antibody tests or immunohistochemistry for that. And I will show later that the fluorescent antibody test and the immunohistochemistry is flawed. I call that the falsification of immunohistochemistry for attempting to identify pathogens within an intact cell, in this case a nerve cell. But here we have combinations mainly of glial cells, and the oligodendroglial cells are here, we presume, the darker ones, and the larger ones more closely resemble astroglial cells. And here there's some evidence perhaps of blood-borne lymphocytes. This particular area we will see is associated with blood capillaries. So here we have in these areas a clearer area, there, there and there. I've called these areas the microinfarct of what? Not to draw attention to myself, but to draw attention to the pathogenesis of these diseases. This is a new concept for neuropathogenesis of these diseases. If this was a human, these are the characteristic lesions that one might find with infarcts associated with cardiac disease, in which there are emboli in these smaller blood vessels that, or even in the capillaries, that produce these microinfarcts. But in this case, with this particular disease, there was no evidence of emboli, but there was evidence of blood capillary endothelial necrosis. And the level of that necrosis reflects the level of the necrosis here in these so-called infarcts, or one might call them some people call them micromalacia. These areas, as you'll see in the next slide, are areas of coagulation necrosis, which is characteristic of hypoxia, which supports the concept that this particular disease is initiated by hypoxia when the blood capillary cells die. So here we have a higher power magnification from the previous figure, and one can see necrosis of glial cells, pycnotic, degenerated, karyorrhexis, that means broken, fragmented nuclei, all characteristic of a necrotic area. Here in this neuropil is necrosis, and some of these cells here you can see are shadow figures or possibly of previous neurons. And so this indicates a much greater severity of hypoxia, a much greater involvement of the blood capillary networks, and a much greater surface area of necrosis, which reflects in this particular lesion, even though it's a micro infarct, it's still a very severe lesion, and it's related not to attack by viruses, as one might speculate, but by the severe degeneration of the blood capillaries of the parent cells. Again, this is a higher power magnification of that area of coagulation necrosis. This was present in the medulla oblongata of a pig, from which we made the original isolation of the virus. A pig showing high fever and a pig showing rather severe nervous changes clinically. So again, we can see the pycnotic nuclei, the area, the eosinophilic area in H&E, hematoxylin and eosin, with the shadows or outlines of the cell, but they're necrotic. Again, this is a higher magnification of that microinfarct of Watt, showing the condensation here characteristic of degeneration of the nucleus, which can lead then to fragmentation. The background is rather an amorphous, structureless in a sense, but still retaining its overall structure without producing a necrosis of a form of liquefactive necrosis where there's dissolution of the tissue. The architecture, by and large, is retained, but it's still necrotic. And it's characteristic, I emphasise, characteristic of hypoxia. And one will see this in hypoxemia, where there are emboli in human cardiac cases, where emboli block the blood vessels. It's a similar sort of reaction. It's the lack of oxygen more than the actual lack of blood, or equal to it. So it's a different concept in the sense that there's no emboli, but the outcome is very similar because of the massive numbers of blood capillaries that become necrotic due to the invasion of the virus. The evidence supporting a new concept in neuropathogenesis of Teschen-Talfen disease and also other related disease of man, including human poliomyelitis. The current dogma or entrenched belief for human poliomyelitis is that the virus directly attacks the neuron and so produces the clinical signs because of the direct attack. The evidence produced here in the Watt pig model contradicts that particular view. The current entrenched view is based upon speculation rather than evidence. The only evidence that's been found from time to time is by the use of fluorescent antibody techniques, immunohistochemistry, to attempt to show that the virus is present within the neuron. I will show later that that is impossible, that the antigen-antibody reaction cannot occur inside that cell because the antibody and the dye have a molecular weight above 10,000, and therefore it's impossible for them to penetrate through the cytoplasmic membrane into the cell. So therefore any supposed positives must be non-specific and flawed. And that's the reason why I did not use immunohistochemistry, for that reason, and because when one uses these techniques, one can see the complete morphology within the cell, all the organelles and the like, which you can't do with fluorescent antibody technique, although bells and whistles seem to be a lot more attractive. In fact, they're not in this research. So here is a spinal cord, and instead of, as well as taking transverse sections, I took longitudinal sections or sagittal sections of the spinal cord. And the reason for that is based upon simple knowledge of the neuron anatomy, where the blood vessels penetrate come into the spinal cord, and therefore if you cut transversely you have a slight chance of cutting through the walls, but if you do it sagittally you'll see the lumens. you'll see the lumens of the blood capillaries. And you'll see that there's a lot more blood capillaries affected than one might think. And if you, for example, cut transversely through there like that, you'll miss these. Whereas if you do a longitudinal sagittal section, you'll pick these up. And you will then see much more clearly that these cell nodules are composed of a blood capillary vasculitis, which I will show you. These are the smaller blood vessels here, which are showing perivascular cuffing. These ones here are the cell nodules, and the mystery will be revealed in the next slides just by histopathology, and then supported by ultrastructural studies. Because once I found this here, I then went on to examine this much more closely using ultrastructural techniques. So here we proceed to a higher magnification, illustrating the point of this perivascular cuffing, presence of blood-borne cells within small blood vessels, and diffuse gliosis, accompanied by early degeneration of the neurons. This is not severe, they're not pycnotic, there's no karyorrhexis, so it's possible that these neuron cells will recover. At the moment, they would not be functioning normally, and therefore that is reflected in the ataxia that we saw in the pigs clinically. So, upon recovery, the recovery can occur in pigs like Harold, pig H. So this is diffuse gliosis, this is perivascular cuffing, this is early neuronal degeneration with central chromatolysis. This is a higher magnification in the same place in the spinal cord, and one can see diffuse gliosis, and looking more closely, these smaller blood capillary cells. See them? Blood capillary cells. The endothelial cells are rather swollen here, and there's mononuclear cells present in the lumen. This is a larger vessel, which is a small blood vessel. And once again, there's a diffuse arrangement of glial cells. They're actually orientating themselves around the various blood capillaries. It's not so easy to illustrate, but there are a number of blood capillaries in these areas, and it's quite possible that they're hidden because they're not cut in the right way to reveal the lumens, but one can see the lumens of these to illustrate the point. So in fact, this gliosis, this cell nodule, is reflective of the presence of the virus within the capillary blood endothelium. Another point about this is of fundamental importance, and a concept which is probably never before reported, and that is the proliferation of these blood capillaries. There's more blood capillaries there than one would normally find. So this is indicative of neoangiogenesis. In the last few years, by looking at the literature, one has seen that with wound and wound healing, that angioneogenesis and blood capillary budding occurs as a response to hypoxia. Once again, illustrating that it's the hypoxia caused by the blood capillary endothelial necrosis which is accounting for these changes. So here is indicative of a response of the body by probable capillary budding. And so when this is accomplished, that means partial or complete re-establishment of blood supply. And so this contributes or could contribute to the concept that of explaining how pig H and other pigs recovered. One of the factors could be this, of capillary budding as a result of hypoxia with angioneogenesis and the development of new blood capillaries in that area. So associated with the myelin degeneration, which can remyelinate, one can begin a conception of how these pigs, and in fact humans, can recover given these types of lesions. So the Watt pig model has application to human nervous conditions of this form. Here is yet another higher magnification of this particular lesion, which area in which the spinal cord was cut longitudinally or sagittally. Once again, one can see these blood capillaries, swollen endothelial cells, and the leukocytes inside. They're very, very small, so you will only be lucky to see one cell. Once again, illustrating what appears to be neo-angiogenesis. Neo meaning new, angiogenesis, blood vessel formations. And it's associated with wounds and wound healing, and I have extrapolated and conceptualized that idea into explaining what is going on with these viruses and the pathology with the viruses. I believe it's the same biological, pathological concept. Same sort of reaction by Mother Nature to a lack of oxygen. So here we have what I believe to be neo-angiogenesis, and once again proliferation of glial cells responding to these local changes and partial degeneration. Here we can see small blood vessels, small cells which are pycnotic, and perhaps even some karyorrhexis of some cells where the nuclei are fragmented. But by and large they're very dark, and most of these could be oligodendroglial cells. The ones that are of a clearer cytoplasm are more like astroglial cells, like there, all reacting to the changes in the neuropil associated with dysfunctional transport because of the blood capillary necrosis and dysfunction of transport of nutrients, of water, of other nutrients, in addition to the lack of oxygen, which is fundamental and vital for the mitochondria to function normally. And we'll have a look at mitochondria later when we go to ultrastructural studies and see those changes which support the changes we see here on histopathology. And so we go on this voyage, this journey of discovery, sequentially. We now turn to a chronic case. This pig was non-ambulatory for 3 weeks. The previous figures were from pigs that were showing the more acute phases, pigs that had a high fever, and pigs from which I was able to isolate the virus from each of those pigs. This is a sagittal section again of the spinal cord, and one can see there's a lack of cellular response, quite different from the one you looked at previously. Here the neurons that are present, the perikaryones, are fundamentally normal. Some cases there might be some chromatolysis. There's perhaps a suggestion of diffuse gliosis, but in general one would consider this to be normal. But this is not true. It's not normal. And we'll see here soon fundamental lesions which reflect the clinical signs of an animal that's non-locomotory for 3 weeks. This is the same part of the spinal cord. There's not a terrific cellular response, but these astroglial-like cells are present, indicative of addressing scar tissue and perhaps assisting in withdrawal of any edema which may be present in this area. So this takes time, and the astroglial cells are responsible for so-called cleanup of so-called scar tissue and some edema. So here we have the dorsal and ventral parts of the spinal cord, which on face don't seem to be showing a huge amount of change. But when we look closer, we'll find a lot more degenerative changes than we saw on face value at first. Closer examination will reveal significant and severe changes. Now here is a higher magnification of the area in the dorsal and also ventral funiculi of the spinal cord. See here involvement of the nerve fibres. These spaces are indicative of myelin degeneration. As I said, on face they might seem to be insignificant, but on closer examination one can see that there's significant myelin degeneration. And here these rather inconspicuous but important indicators, which are the myelinophages, quite often overlooked by research workers, but very important to indicate that there is myelin degeneration. Now in the clinical cases, sometimes the myelin degeneration is too severe and there may not be remyelination, but in pigs like pig H, there could have been myelin degeneration in pig Harold, the pig, and subsequent remyelination meant that the pig could recover, along with other recovered cells in the nervous system which were affected. This is taken from a spinal nerve root, a longitudinal section of the spinal nerve root. And one here you can see the normal nerve fibre, and here is abnormal. Notice the increase in cellularity, and when we go on to higher power view, one will see these subtle myelinophages indicative of myelin degeneration. So this is a normal one, and this is an abnormal one, showing the changes which I illustrated before of myelin degeneration. So this is a closer high magnification of the normal nerve fibre, which will be contrasted in the next slide. Once again, this is multifocal demyelination. It may appear to be subtle and not of great significance, but it's sufficient to produce clinical signs. Here we contrast the normal nerve fibre that we saw before with this abnormal one. And one can see at first glance lack of continuous cellularity and a lot of spaces that are present there. Again, this is a fundamental lesion associated with the neuropathogenesis of this disease, which quite commonly is overlooked. One can see these spaces and there, indicative of myelin loss, and here at 2 one can see a residual mass indicative of myelin. The spaces there indicate loss of myelin, and here we can see the residual mass where it's still retained. So these lesions, which are shown here and in the next couple of slides, indicate that they're severe enough to account for the changes that we saw clinically. Again, one finds overall degeneration of the fibre, and here are the myelinophages, and here is an expanded axon cylinder. In many cases, it would appear that the axon itself is not severely affected. There is no obvious damage, segmental or otherwise, of that axon. It is an indication of swelling of the axon, which could be resolved, and with resolution, accompanied with the remyelination, could account for the response positively of the pig, like pig H or other animals that recover, and indeed humans that recover, showing these sorts of changes. There they are, myelinophages, subtle lesions, not easily observed, easily overlooked, but of great importance as a contributor to neuropathogenesis. The diagnostic histopathology of this disease revealed that there were severe lesions not only in the central nervous system but also in the peripheral nervous system. With regard to the peripheral nervous system, the lesions were found in the dorsal root ganglia and their fibres, as I've pointed out, their nerve roots, and also in the trigeminal ganglion, the Gasserian ganglion, which was equally affected. And those lesions reflect the nervous changes that are seen. In fact, the lesions of the dorsal root ganglion could be a major manifestation or reflection of the changes seen clinically. So here there's a marked proliferation of the satellite cells associated with the very large ganglion cells present in the dorsal root ganglion. Now, the ultrastructural studies were done for the very first time in the history of this disease. Ultrastructural studies were done on the dorsal root ganglia, and the studies were done there to uncover the pathogenesis of the disease at the ultrastructural level. So this is a dorsal root ganglion, and it's showing glial cells, and it's showing in satellite cell proliferation around and now we continue with our voyage of mystery, attempting to isolate the pathogen. We have narrowed the story down to the most probable etiology as being a virus. And for this reason, we've taken samples as a surgical aseptic method from the brain and spinal cord and the dorsal root ganglia and Gasserian ganglia at various times, and attempted to isolate a virus on tissue culture. And this illustrates the first isolation of porcine teshovirus 1 in Australia on tissue culture. This here is a normal monolayer of tissue culture. The tissue culture that we used was fetal pig. We obtained those fetuses from the local meat export abattoirs, and using aseptic techniques produced the monolayer. We also had a laboratory persistent tissue culture which we also use, which is known as pig stable cells, and we had another line called pig stable equine kidney cell line. And on all of them we isolated the virus for the first time in Australia, and I might say for the only time since that time in 1970, which is 44 years ago. The virus has never been reported as isolated since, which in some ways might appear to be rather remarkable, but I'll demonstrate how we did it in the following slides. One of the most important things in tissue culture, as virologists know, is to avoid contamination, and we were ultra careful in doing that. It's very easy to get contamination, and therefore that will ruin your results. So I don't know what other virologists have done, but when we did it, it worked well. So here is the normal tissue, and here's the early stages of indicating the presence of the virus, where the cells lose their attachments to each other and break off and become round, rather refractile and shiny. So here's a more advanced stage in which there's lysis of the cells, refractile, they lose their attachments. So what we do to identify the virus is to take a sample at this stage or just before, and we use what's known as a cell lysate technique. It's a very simple but very effective method in which we just add a drop of water to the cells, and they take the water in, and being semi-permeable, expands, bursts the cells, and releasing these almighty virus particles. It's a beautiful and simple technique for identifying the cells. They are about 25 nanometers in diameter, icosahedral in formation, and they appear to be surrounded by, they are surrounded by a membrane which could be smooth reticulum. So we have a positive identification of a virus in pure form. So what we'll do is to characterize that particular virus. One way to do it is the use of neutralization tests, and we'll purify the virus and then transmit it back into the brain of recipient experimental animals to once again reinforce this concept of Koch's postulates adapted to viruses from bacteria work. The method of proceeding with virology is to use the dilution methods in tissue culture, a 10 by dilutions we used, log 10, and the whole idea is to dilute it right down to N dilutions, so we've got a purified number of particles there which we can grow again, inoculate into the animals. Another method of demonstrating that this is a porcine Téchaux virus, one, is by the plaque technique. It's positive for plaques, and it's also a method for getting to N dilutions to purify the virus. So these next few slides just illustrate that it was plaque formation positive. The first one is a control, which is normal, a normal tissue there. So that's a control with equine kidney cell line. The next one shows the lysis in parts at a dilution of 10 to the minus 3 for equine kidney cells. This was taken from pig virus 1261. And this final plaque formation demonstration is at 10 to the minus 4 dilution, indicating a few areas, a more N dilution type of method. And from these we can presume that we're getting a more purified virus by the N dilution method. But the main purpose of this is to indicate that this virus produces plaque formation. We're continuing with our journey of attempting to prove that this virus is the etiological agent. So the next step after purifying it on tissue culture is to trypsinize the cells and take the samples at highest dilution, 10 to the minus 6 we used, highest concentration, and we inoculated that into the brain as I illustrated before, and you've already seen the motion picture videotapes of this particular pig, which showed the characteristic nervous signs in the natural outbreak, which reinforces the concept or confirmation that this virus was indeed the etiology. One emphasises this greatly because in the research literature, quite often the importance of the etiology is overlooked. And a glaring example of that is stultified research on Alzheimer's disease for 25, 30 or 40 years, where it was almost accepted by the research community that amyloidosis was the cause, almost the etiological agent. So people did not go, research workers did not go looking for the real etiology. So I emphasise this because so much time and effort and millions of dollars was spent on searching for things like immunoglobulins to amyloid, when amyloid is just a secondary glycoprotein product of biochemical pathology. So instead of going and looking at places like blood capillaries and looking at the pattern of the disease like I've illustrated here, one just got the secondary product and went overboard on it. So in the past with Alzheimer's, there's been some evidence that the blood capillaries are affected, and they may well be the fundamental lesion, but in many cases this research, which is quite legitimate research, has been overlooked in the favour in favour of flavour of the month, flavour of the decades, amyloid. It now transpires that amyloidosis and the whole concept of it is run into a dead-end wall, and so one is left with Alzheimer's disease of not knowing really where to go. And so one might then look towards these fundamental what concepts, the pig model, to go looking for the fundamental lesions that are present in diseases like Alzheimer's disease. The use of topographical mapping, qualitative and quantitative, the significance of looking after you've seen the pattern to decide where the lesion is located. I strongly suspect that it will be found in the blood capillary, and I strongly suspect that the mitochondria will be affected as well. And my postulation with Alzheimer's is that this is an epigenetic condition with mutation of the mitochondria as an aging process, and I don't know exactly what that is, but it seems as though there's some sort of mutation of the mitochondria or associated organs which result in aberrant biochemical metabolism and the deposition of deposits which are not normally there because of the dysfunction of the biochemistry. So once again, emphasise that it's absolutely vital to have a systematic approach when doing research, and rather than fly-by-night, hit-and-miss method, which is supported by millions of dollars for research, and the evidence so far shows that it's been wasted money. The evidence so far shows that the situation is going from bad to worse as the population ages. The situation shows that it was 1907 that this disease was first described. Some years later there was amyloid found, but nothing else has advanced. Nothing else has advanced. And so it's of critical importance that we return to fundamental, systematic neuropathology and neuropathogenesis in the way that has been described so far in the Watt pig models. So here is at day 10, the pig, as you recall, showed some slight changes in ataxia. The next day, on day 11, there was an increase in the severity of the nervous signs, but not greatly severe. The animal could take weight on its front legs, but there was crossing over here of the hind legs, as you saw on the video. And the next day, things got worse. On day 11, the changes became more severe. Here's the animal held up here. It still can stand, but when let go, it shows evidence of rather severe posterior ataxia, weakness. Pig, when unsupported, drops down. It can get up if supported, but shows severe posterior paresis of a more flaccid type, not spastic like the tetanus cases. This is day 12. The animal is more severely affected. It can walk on its front legs if supported posteriorly, but this side and the next one, it's more severely affected on day 12 than on day 11. At this point, this animal was destroyed and the samples were once again taken from the brain, spinal cord, dorsal root ganglia, and the virus was successfully re-isolated from that, so proving the positive nature of the transmission. This particular work stands in stark contrast to the work that's been done, or the lack of it, on Alzheimer's disease. Unfortunately, there's only been one attempt at transmission for Alzheimer's disease. A number of brains were taken and they were attempted transmissions were made into chimps, and out of all that, after a fair period, one animal showed nervous signs. It would have appeared that the materials and methods in that research paper were immaculate, but then the authors refuted their work quite surprisingly, and it would appear that they did not get the lesions that they wanted. So they then refuted their own work, saying that it's highly likely that there was cross-contamination. One of those authors was a Nobel Prize winner, and the other authors were very prominent in various Alzheimer's disease institutes. It's unfortunate that there's only been one attempt at transmission for this disease, because if it is indeed transmissible, it changes the whole complexion of treatment, control and prevention. And along with topographical mapping, which I mentioned before, qualitative cross-sectional mapping, and more urgent examination of the fundamental lesions, coupled with more repeated attempts at transmission, this disease could be solved. Unfortunately, the systematic fundamental research on this disease has not been done, and it's most unfortunate that this has happened. But let's hope that with the use of the Watt pig models or other models which are rather similar in the future, that this disease will be solved. Because at the moment, all that's offered to the world is things are going to get worse rather than better as the population ages. If we don't find the fundamental reason for that lesion associated with aging, we're going to be faced with an epidemic of people who need special health care. The cost of that and the waste of human beings in not only the carers but also the patients is immense, because quite often this senile dementia occurs at quite an early age, even 40s or 50s, and the fundamental neuropathogenesis remains unknown for most of these diseases. And so it's time for a change in concept, a change for the better, a change for a more fundamental, systematic approach to these critical diseases. In the previous work that we've looked at, the natural outbreak showed a range of nervous signs. This indicated that the host resistance and the host immune mechanisms must have played a role to account for this variation from pig to pig, given that the virus was exactly the same. So it was I decided to have a treated group with cyclophosphamide and virus, and group with virus only, both of them inoculated intracerebrally, to attempt to achieve two goals. One, examine the immunology, and two, conduct a sequential study, more closely examine the neuropathogenesis as it developed. On the left-hand side here is at day 6, one sees the characteristic Watt microinfarct, as I call it, with a reaction there of glial cells, many of them dead, because this is, once again, this is coagulation necrosis. On this side, with virus only, this is a cell nodule with many degenerated cells. This is the medulla oblongata, and this is the spinal cord. So here we have a mixed population of cells again. These spaces indicate edema, and these are the glial cells and possibly the lymphocytes. And here is some disintegration of cells, some of which are typical of myelinophages. Here and here, these cells are not unlike myelinophages. So here we have edema, and we have evidence of some focal demyelination of the spinal cord. So here is the olive nucleus in the medulla oblongata, showing central chromatolysis, early degenerative change, but reversible. Here is the spinal cord again, showing condensation of the nucleus and diffuse type of chromatolysis, and this is unlikely to recover. It's considered to be an irreversible change. This is the Gasserian or trigeminal nerve ganglion in the virus only, and in general the Gasserian ganglion and the dorsal root ganglia were affected in a similar sort of way, and quite severely, with degeneration of the ganglion cells and with myelin degeneration. Here is an infiltration, large numbers of cells. Whether this is lysis with degeneration of the neurons is uncertain. it's not possible. All I can say is that there's a large accumulation of cells, of glial type cells and blood-borne cells, mononuclears in this region. But it's not possible on here to say whether there's actually lysis or not. It may and may not be. But lysis is not really characteristic of this particular disease because the neurone itself undergoes hypoxia rather than invasion by a virus, which tends to produce lysis. Here we can see a swollen axon, and here are these karyorectic-like structures which are identified as myelinophages. So we've got nerve fibre damage, the axons are swollen, the myelin is degenerated, and there's a cellular reaction attempting to clean up the debris, with the possibility of replacing it with normal at a later date. Here is still with virus plus cyclophosphamide. The axon cylinder is enlarged, and inside it are these myelinophages there, and these are indicative of myelinophages as well. So again, nerve fibre degeneration and myelin degeneration with myelinophages. We now proceed to day 8, and this is virus plus cyclophosphamide and virus only. This one here is from the neuropil of the medulla oblongata, and this one here is from the lateral vestibular nucleus in the medulla oblongata. Axon swelling of the cylinder around here, and the eosinophilic nature of the axon. It's not eosinophilic here, but it was on Haematoxylin niacin. On the right hand side is the lateral vestibular nucleus in the medulla oblongata, where there is a cell nodule here and perivascular cuffing here. Here as well is evidence of neuronal degeneration associated with the cell nodule, which I call the cell nodule of Watt because I think it's important to attract the attention of research workers to the place where the virus is likely to be in the blood capillary endothelial cells. So here is the virus plus cyclophosphamide with a swollen axon. Here a swollen axon cylinder surrounded by these glial cells. Some of them look like oligodendral glial cells, and those with large foamy cytoplasm nucleus are the astroglial cells. Again, this is from the medulla oblongata. This is a higher power view of the previous slide with virus only, and this is from the lateral vestibular nucleus in the medulla oblongata, and one can see irreversible degeneration of the neurons, the perikaryon. Nucleus is pycnotic, cytoplasm is breaking up, vacuolated, and here is those spaces, evidence of edema, and these cells here accumulating around what will be capillary vasculitis. What is capillary vasculitis is a cell nodule. rather diffuse but more concentrated in that area. One can't quite see the lumen of it, as we've seen with sagittal sections. That lumen is present. And so this is the place where the virus is likely to be, producing the blood capillary endothelial necrosis, which then has an effect upon the surrounding cells in the neuropil, producing dysfunctional transport of nutrients, including hypoxia. And these neurons are not showing invasive virus, but they're showing the effects of the neuron, the effects of the blood capillary necrosis and dysfunctional transport systems, particularly the lack of oxygen, which we'll see later is affecting the mitochondria. A number of these cells here are rather pycnotic, these glial cells, as though they too are suffering from the effects of dysfunctional transport, particularly hypoxia. So they're rather pycnotic, small, shrunken, and so this is characteristic of an area where the virus is operating and producing these effects in the neuropil. So here with virus on day 8, we're still on day 8. These are normal neurons showing the Nissl substance, and this one here is showing severe degeneration. So you're getting multifocal changes, sometimes just subtle single neurons affected, in other cases more affected. The severity of these lesions, whether they be the neurons or the other cells, is dependent upon the mortality rate of the parent blood capillary endothelial cells. This concept is completely original. These cells here, again in the medulla oblongata, are showing increase in number, glial cells, reacting to the presence of myelin degeneration. The spaces are likely to be edema. These various glial cells are reacting to them. Again, this here in 77A is the medulla oblongata, as this is too, with virus plus cyclophosphamide, and here with virus only. Here we can see the spaces indicative of edema and myelin degeneration, and here are characteristic myelinophages, which are phagocytizing the dead myelin. Around here, various glial cells. The darkly staining ones are called oligodendral glial cells, and these ones here are astroglial, which have the function partly of taking up the edema in the region. And here is myelin degeneration with the edema, and here is a myelinophage within the axon, within the axon cylinder. I beg your pardon, we go over here, we'll see there's a lot of karyorrhexis in that area, and that's indicative of an infarcted microinfarct part. So one can see here evidence all the way through of quite significant white matter degeneration, whereas the literature states that this is mainly an effect upon the grey matter, whereas in fact I'm demonstrating here considerable evidence that there's white matter change, And of course, if there's white matter change, there's going to be changes in the myelin and the like, and the nerve fibres, and change which is manifested in the clinical signs. This is strong evidence of myelin degeneration. So now we turn to the experiment which I referred to before about inoculating pigs intracerebrally with the virus and separating them into two groups, one which was inoculated intraperitoneally 24 hours after that inoculation with cyclophosphamide. Cyclophosphamide was injected intraperitoneally at 60 milligrams per kilogram, which we found out to be the optimum dose rate that was not lethal but suppressed lymphocytes. So the results of that exercise, that experiment, were that in all animals, all 12 animals, there was a fever, 40 degrees C and above. And in all except one, there was some degree of nervous signs. In the group that were inoculated, the 6 that were inoculated with virus and then the cyclophosphamide 24 hours later, they showed severe changes characteristic of the classical Teschen disease that had been reported in the early 1930s in Czechoslovakia and probably in Poland, and had spread during the late 1930s into Germany, producing high mortalities and high morbidities. Those animals, those 6, showed quadriplegia. The others were showing more of a hemiplegia, the posterior paresis, paralysis. And so using the single virus for that, and the same virus for the virus only, I reproduced a disease which was a classical Teschen disease. This is original research work, and what it does illustrate is the impact of the immune response upon the clinical signs, something which I suspected at the beginning when we got a variation in the nervous signs in the natural outbreak, even with with the single virus, which we isolated, purified. There were no other viruses, and there was no indication whatsoever that there was a virulent virus and a milder virus. They all proved to be the same. When they were inoculated intracerebrally, they produced a Talfan type of disease. In the original natural outbreak, there was only 1 pig out of 100 that died. And that pig showed the characteristic changes of a classical Teschen disease, which once again indicated to me that it depended upon the immune response rather than the, I don't know whether you call it cult, the current dogma or entrenched belief that there are 2 strains, the Teschen virulent one and the milder Talfan one. Because the bladders are enlarged in all of these, we can immediately say that there are severe lesions associated with this porcine Tesho virus above T6, because that's the sort of pathology that's produced in this disease. Now, these neurogenic bladders and dysreflexia, autonomic dysreflexia, have never been reported in the literature, and to draw attention to them, I've called than the neurogenic bladder of what? Not to draw attention to myself, but to draw attention to the pathology, so that in the future, after 100 years of looking at Teschen disease, we finally might find that there's dilated bladders in the more severe forms. For example, there's a severe outbreak in Haiti. There's no reports at all about the dilated bladders. In fact, there was no reports at all about whether blood samples were taken, and in fact, only the virus isolation was made. So people come to the conclusion, the wrong conclusion, that there's an introduction of a new virulent strain. This strain is stable. This virus is stable, and therefore, when there's an outbreak of a disease as serious and as severe as Teschen, one should also think of a possibility of a natural immunosuppression which is eliciting the severe changes compatible or comparable with what I've produced here with a synthetic reproduction of it. Now, those sorts of agents which can immunosuppress are considered to be, for example, circoviruses, and the possibility also exists that the AIDS virus might have got into the pig population in Haiti. Originally, the AIDS virus came from monkeys, it is said, and got into the human population. There's no reason why it shouldn't get out of the human population and share the disease with pigs. If it did, then the same sort of pathology would be seen, in which there's an attack upon the lymphocyte in AIDS, producing widespread immunosuppression, and the humans die from what were rather innocuous infectious agents under normal conditions. One might postulate that AIDS could be there in those pigs. The other thing about this whole issue is, once again, the development of the neurogenic bladder, where it's greatly dilated, producing autonomic dysreflexia, and in humans, unless treated, can lead to death. So what I'm postulating is that in Haiti, where that recent outbreak occurred, and the outbreaks that occurred in Europe in the 1930s may well have been associated with natural immunosuppression from concurrent viruses, such as circoviruses. I say this because in my hands and in the hands of other people over the last 30, 40, 50 years, attempts to reproduce the classical Teschen disease have largely been in vain. The so-called virulent strains, when injected, quite often produce nothing at all. One might say it's innocuous because of passaging, but it also, the issue is that immunosuppression does play a significant role. It's the cervical spinal cord showing rarefaction of the dorsal funiculus. The axonophages here in one, and they're in swollen axon cylinders, which is 2, and there are densely staining, nuclear staining of oligodendroglia in 3. So 79A is the same as 78A, higher power view. There's a degenerating axon in 1. There's a axon cylinder surrounding it in 2, and there appears to be an axonophage in the middle of that degenerating axon. In 78B, which is from the medulla oblongata, in the lateral cuneate nucleus, and there are pycnotic nuclei of suspect oligodendroglial in the nearby neuropil. So this is the central chromatolysis in the lateral cuneate nucleus in the medulla oblongata. 79B is the neuropil near 78B, showing malasic foci, edema and demyelination with spaces due to dilated axon cylinders and or edema in 1, and characteristic myelinophages myelinophages in those spaces. Again, indicative of white matter destruction. So it can be seen from here that there's considerable white matter destruction in both the virus and the virus plus the cyclophosphamide. We now turn to day 11, and 80A is the virus plus cyclophosphamide. This is the nucleus of the facial nerve. There are spaces of various sizes, mostly due to axonal degeneration, but perhaps some are spaces left by myelin degeneration. In virus only, this is the white matter of the spinal cord. There's a dilated axon cylinder containing suspect axonophage, is virus plus cyclophosphamide, and this is the epithalamus. There's a cell nodule spaces there. This could be edema and part of the white matter degeneration containing these myelinophages here at number 2. Virus only, and here we can see the beginnings of degeneration of the neurons, but one has to be careful here because the pericardium might be just cut off at a particular pace and it gives the appearance of degeneration. Day 11 post-inoculation was the finish for all of the virus plus cyclophosphamide. The last one was showing severe quadriplegia and was comatose and was destroyed at that time. The one left is the day 17 post-inoculation. 82B is the spinal cord showing vasculitis here at 1 and increased glial cells, diffuse glial cells over the neuropil. In 83B is a higher magnification taken at the medial part of the dorsal funiculus. There's swollen eosinophilic axon here at 1 within a dilated axon cylinder here at 2. Some contain axonophages. Some of these might also be myelinophages. It's not really possible to distinguish them with absolute certainty. 84B, it's similar to 83B, and here we see evidence of the axon cylinder swelling, two, the axon cylinder, and one is the axon itself swollen with myelinophages. So this again is from at the medial part of the dorsal funiculus. Skin healing, which is not exactly part of the neuropathogenesis, but is put in because of interest. This is skin healing at the site of the intracerebral inoculation of the virus on day 11 post-inoculation. With virus and cyclophosphamide, one can see retarded skin healing. There is epithelial regeneration here, along this line here, and it is proceeding under a purulent scab area. This is a higher power view of that same area, showing epithelial downgrowth here, some macrophages here at 2, and a few lymphoid cells, 3, and some fibroblasts at 4. So one can see at this stage that the cyclophosphamide has worn off, has regressed, and there is some cellular reaction returning to the area. This skin healing is not as good as the virus only, because here we can see with the skin healing virus only is more advanced than 85A, and there's re-epithelialization almost completed. And a high power view of that virus only shows a lot more fibrocytes than 86A and a few lymphoid cells. So this is put here to indicate the effect of immunosuppression upon skin healing. Here on figures 87 to 102 is the visual evidence critical to the thesis on neuropathogenesis. These are the ultrastructural studies that were done using porcine teshovirus 1 in experimental pigs. The first thing is that there was total body perfusion using 4% glutaraldehyde. The animal was anaesthetized and the fluid was injected into the heart. The body stiffened and there was yellow pigmentation from the glutaraldehyde throughout the whole body, indicating effective perfusion. In this way, there's no doubt that the very sensitive organelles such as mitochondria did not suffer from post-mortem autolysis. The other evidence to prove that there was no post-mortem autolysis was that there were normal parts and abnormal parts in close proximity. If there was post-mortem artifact, there would be just a general change which shows up on histopathology as eosinophilia with H&E slides. So one is quite confident that all the changes that are seen here are in fact true and not post-mortem artifact. The following electron micrographs of ultra-thin sections of dorsal root ganglia were selected from normal, non-immunosuppressed, and cyclophosphamide immunosuppressed pigs in this experiment. So this is taken from the dorsal root ganglia, and there has been no previous experimentation on the dorsal root ganglia with this virus, porcine teschovirus, and not on other related pig picornaviruses, nor on human picornaviruses. So these are unique findings which could have application to both man and animal. The dorsal root ganglion is a little different in its blood supply from some other parts. Because of the ganglia, there's a complex and considerable blood capillary network, and there are gaps in the blood vessels which allow closer contact of the blood with the contiguous other cells. These pigs were destroyed sequentially after inoculation with the Queensland isolate of porcine teschovirus 1. We turn to the first of the figures. Except to note that the cell nodule, which is mainly focused on here, was identified on a light microscope using toluidine blue stain. Then sections were taken for EM from there using the technique of Grimley. So turning to figures 87 and 88, day 6 post-inoculation, virus and cyclophosphamide. Before turning to that, one must say that for the first 4 days there was no fever. On the 5th day there was fever in all of the pigs. One can interpret that fever as being a reflection of the blood capillary necrosis with the voiding of its contents into the bloodstream, producing pyrogens, and therefore fever. The whole issue of production of fever is a highly complicated biochemical complex. But one of the important things is the presence of various pyrogens which elicit the fever. So therefore, fever can be seen as an indicator of degeneration of the blood capillary endothelial cells, which is the fundamental lesion of neuropathogenesis in porcine teschovirus 1, and indeed a number of other viruses of animals, and in man, including human poliomyelitis. This evidence is contrary in the entrenched belief for a number of viruses of man and for human poliomyelitis, where it is still entrenched in the belief of research workers that human poliomyelitis virus attacks the neuron directly. But turning now to day 6 with virus plus cyclophosphamide, this particular animal at the time did not show any clinical signs, but one took it so that one could see the earliest possible lesions. And it was fortunate that this was done, because it can be seen that the lesions is well and truly advanced by day 6. And day 5, where there was a high fever, but no nervous changes at the time, is a reflection of the severe injury that one can see here. So on figure 87, this arises from the cell nodule, and this is a degenerated oncocytic cell showing cytotoxic edema, this here. And by the look of it, there are other cells that also degenerated in adjacent areas, contiguous areas. So here and here and here could be seen to represent separate cells that are degenerated at the time, at day 6. Here is another cell which is actively reacting. Its ribosomes are freed and it's very active, and it's showing the beginnings of these villi here. Here are active leukocytes, and they're showing villi or pseudopodia-like formations, and there is considerable amount of rough endoplasmic reticulum. That C is a normal satellite cell which surrounds this normal neuron. So one can see at this stage that the neuron is normal. It's not certain that these blood capillaries supply oxygen to this, but it does underlie the point that at that time no nervous signs were seen. This whole picture raises a considerable, interesting and original and new concept of neopathogenesis with regard to these viruses that invade the blood vascular system. The entrenched belief, the predilection sites for any virus, that is that there are specific chemical receptors. Unfortunately for this hypothesis, the specific receptors are not really specific, in that the lesions don't occur in some places where there are so-called specific receptors. So looking for another explanation, one can find it by conceptualizing on differential physicochemical blood flows at the blood capillary circulation site. By applying just basic principles of physics on surface area and friction, one can see and visualize is likely to be happening in this invasion of virus. When the virus gets into the bloodstream, if there were no cells at all, if it was acellular and it was just fluid, one would expect that the virus would be diluted out. And therefore, by logical deduction, one should expect that those viruses become attached to the circulating blood cells, some of which are these lymphocytes. And because of the interaction of lymphocytes with with the virus in terms of immunology, then it's likely that the lymphocytes are the ones that are affected. But at the early stages, it's quite possible that the lymphocytes have not been conditioned enough by dendritic cells and macrophages which present the antigen to them so that they can kill the virus. And so we have that immediate cell, a mediated response of T cells and K cells. And this is probably some of them. It's only later, 10 days, 14 days, maybe even 3 weeks, that the B cells convert to plasma cells and produce the antibody. So therefore, the immediate effect is a cell-mediated response from principally T cells. So here we can see villi, beginnings of villi here. Here we can see these pseudopodia here. So it would be easy to conceptualize that this increase in surface area will decrease the blood flow. And the other point is that the blood capillary networks are more complex in higher areas of energy requirements. And therefore the surface area, total surface area of those will be higher than in other places. So here we have the combination of the cells producing friction, blood-borne cells, and we have the blood capillary endothelials reacting, somewhat like fixed phagocytes, in that they react to the presence of particles. And after all, it would seem that rather than a chemical reaction, it's a physical reaction to these virus particles. And so it's obvious here that the surface area is increased, and when these increase, and we'll see later capillaries how they're markedly increased, there is a general slowing down in the flow of blood, a greater random opportunity for those viruses to be attached here to the blood capillary cells. Even at day 6, we can see that there is liquefactive necrosis of these blood capillary endothelial cells. Obviously, when this happens, there is going to be total dysfunctional transport of nutrients from here to here. Normally, the purpose of these blood capillaries is to transport nutrients from here into the surrounding neuropil. Quite clearly, this is completely dysfunctional in terms of the Golgi apparatus, which is concerned with transport. The Golgi apparatus will be severely affected. With the lack of energy, the Nissl substance is likely to break up. Any bonds that are present there need energy support, so they'll break up. So one will see, rather than the clumps of Nissl substance, you'll see free ribosomes distributed throughout the perikaryon. And the other vital thing, of course, is the mitochondria. And here they're basically normal. This is a fundamental lesion which marks the initiation of pathogenesis for porcine tissue virus 1, which has never been recorded before. Figure 88 shows part of a perikaryon. In general, it's fairly normal, The mitochondria here are within normal limits, and the Nissl substance is present in clumps, like in the previous figure. The hypoxia does not seem to have taken its effect as yet, certainly in this particular one. But there's an increase in polyribosomes here. There's an increase in free particles, single particles. For A, there are also less dense particles here in C, which may in fact be glycogen particles. All of this is the beginning of a dysfunctional transport system. There's an overall impression of activation of the Golgi system, and the Golgi system is critical in the transport system of nutrients throughout the cell in a very, very complex manner. So that was day 6, virus plus cyclophosphamide. We now turn to figures 89, 90 and 91, and this is day 6 post-inoculation, virus only. Once again, one can see the fundamental lesion in the pattern of this disease. And here in A is once again a swollen, it's called oncocytic cell, which has taken in water because of the increase in permeability of its cytoplasmic membrane because of injury. And it's almost devoid of all organelles. And so this is known as an oncocytic cell with cytotoxic edema. Next to it appear to be other cells that are also degenerated. Here, and also most probably the remnants of another one here. As one can see, this liquefactive type necrosis is identical with that of the immunosuppressed animal, virus plus cyclophosphamide. And so it is indisputable that this is the fundamental lesion. Before, we noticed in the previous figures that there was like pseudopodia or villi in the leukocytes. And here we can see it in a thrombocyte. So it would appear that the virus stimulates reaction not only in the leukocytes but also in the thrombocytes. And so the thrombocytes have villi which produces increased surface area, which produces increase in friction, again slowing up the blood flow and so permitting random attachment, entry, invasion of the local blood capillaries endothelial cells. The remains of the cytoplasmic organelles here in A appear to be ribosomal material, all detached and free. In here, the contiguous one is the completely devoid. From this evidence so far, both here and back here in figure 87, one can develop a hypothesis on the spread of this particular virus. One has alluded to the method by which the disease is initiated by physicochemical means, slowing of the blood flow, and one can also develop, based upon visual evidence here, a hypothesis that the spread is by systematic, contiguous virus attack on the blood capillary endothelial cells. So here are the cells which have been destroyed. If virus is released here and close to this capillary endothelial cell, it is possible for that virus to become attached and enter it. And one can see here the reaction that one can see on this particular cell. It's very active and there are lots of free ribosomes there, and it's quite likely that this one would have eventually been invaded by the virus. It may have already, it's uncertain, but this underlies the spread, the cause or the explanation for the cause of the spread. In the absence of T cells and B cells, the pathogens are free and available to continue spreading. The vital cell is the T cell in the early stages. If it's lacking, then that permits this continual spread from cell to cell. And this explains why the lesions are more extensive and therefore the clinical signs are more severe in the immunosuppressed animal compared to the non-immunosuppressed animal. In the non-immunosuppressed animal, the clinical signs are of a moderate change of paresis to more severe hind leg paresis. The clinical signs in the immunosuppressed is one of quadriplegia, an inability to rise and involving both the front legs and the hind legs, coupled with nystagmus, head movements and body movements, shivering. So in the research in the early stages, using the oral route and also by natural transmission on the deep litter system, revealed that there were lesions throughout the spinal cord, but the clinical manifestations was only demonstrated in the hind legs. This again might be related to the complexity of the capillary network with subsequent increase in surface area and frictional forces, which I've just alluded to. I'd also like to point out on 89 that this here is another blood capillary. Although it's fairly normal, it does seem to be quite active, and it's quite possible that that will be affected in the scheme of things. But it's obviously much more normal than this one here. So this is a sporadic, to me, randomized type of disease spread. Here in figure 90, day 6, post-inoculation, one can see here at A degenerated satellite cell, an amphicyte, which is adjacent to perikaryon, which is showing early stages of degeneration. The satellite cell contains almost no organelles. The perikaryon here shows vacuoles of various sizes, and at least some of these vacuoles appear to be damaged mitochondria. Here at C, again, is a normal blood capillary endothelial cell. Figure 91 is a high power view taken in this area of 44,000 times magnification, showing the severe reaction in the perikaryon, which is characteristic of hypoxia. These lesions in the mitochondria are characteristic of hypoxia in the sense that past workers have produced anoxemia by cutting off the blood supply and reproducing these types of lesions. So therefore, it's not caused by primary attack of the virus upon the perikaryon. It's caused by secondary hypoxia. In this particular condition, obviously with porcine virus 1, there are no emboli. There's no cutting off of the blood supply. There's no hypoxemia, but there's oligoemia, a reduced amount of blood, primarily because the blood endothelial cell is necrotic, and therefore the nutrients that would normally pass through it to the surrounding neuropil are not present. Those nutrients include food, gas and water, and those are not forthcoming. These lesions here are characteristic of what's known as oxidation of the mitochondria, and the other name that's given to it is oxidative stress, because the mitochondria depend upon the oxygen. Obviously, with sufficient oxygen, they have aerobic respiration where they produce 36 ATP, whereas in anaerobic situations they do not. The ATP is reduced in amount, so the function is reduced. Not only does that occur, but there is this condition known as free radical peroxidation, in which the free radicals bouncing around actually destroy the surrounding chemical complexes that make up the mitochondria, and so therefore the cristae and the like are destroyed. Some research workers have produced evidence that this type of condition is caused by reperfusion. When they reperfuse the area after blocking it off, they get these free radical stress conditions. In this particular condition here with porcine virus, reperfusion does not seem to be a factor, but the lesions are similar. Therefore, the statement is made here, supported by evidence, that reperfusion is not the only cause of mitochondrial damage. It is self-evident that this is not happening. As far as the rest of these areas concerned, there's a considerable increase in free ribosomes, brought about because the Nissl substance would be freed. The ribosomes in the rough endoplasmic reticulum would be freed up because it would depend upon energy that comes from normal mitochondria.
So that accounts for the dispersion and release of the ribosomes. Behind that is a background of markedly increased activity of Golgi apparatus, what appear to be smooth endoplasmic reticulum cylinders. All of this associated with a dysfunctional Golgi apparatus system. We now turn to figures 92 to 98. Day 8 post-inoculation, virus plus cyclophosphamide treated pig. This particular pig on day 8 showed a fever of 40.7, 40 degrees is fever, and it showed severe quadriplegia. This figure 92 shows in a degenerating blood capillary endothelial cell next to one that is beginning to be affected. In a virus characteristic virus crystalline array within the cytoplasm, which is pathognomonic for the etiology of this disease in the blood capillary endothelial cells. These blood capillary endothelial cell virus crystals, each at approximately the same size as that which we saw in the tissue culture of about 28 nanometers. Here one can see the depletion of organelles, some evidence of rough endoplasmic reticulum. Mitochondria are there, the ones that are normal are there. And here, along the cytoplasmic membrane, we can see that it's undulating, it's reacting to injury, and some villi are here that are cut off. Once again, this surface area was increased, permitting the random attachment and invasion of the virus. This one at B is not normal because it's showing a reaction to the presence of the virus and or the presence of those adhesive leukocytes and thrombocytes, the sticky cells, increasing the surface area. Here is a mitochondria which is very large and indicates that it might be showing hypertrophy as a reaction to injury. And here's another one which looks reasonably normal, another mitochondria which looks reasonably normal. In here the cristae are still retained. But the organelles are disorganized, as one can see, and there will be dysfunctional transport systems of the Golgi system, and in fact the whole of these systems which brings the nutrients from here in the luminal surface to the abluminal surface and then into the neuropil here. So here in A, with the virus plus cyclophosphamide, is the presence of virus crystals. In C, which is a pericyte surrounding the blood capillary endothelial cell, there's complete depletion. This indicates that the virus has attacked, invaded, and produced complete loss of organelles in this system. On this view, there's no virus crystals. Virus crystals would be a hit and miss operation. And here we're lucky to see, fortunate to see the virus crystals. But later on, in another figure, I will show you the virus crystals in this pericyte. Once again, pathognomonic for the disease. Now here in D, by and large, this perikaryon is normal. The mitochondria are normal. Maybe they're a little enlarged, but they're normal in the sense that the cristae are still there. There is some indication that the rough endoplasmic reticulum, the nissl substance, is starting to break up and producing free ribosomes throughout the cytoplasm. Here in figure 93, which again is virus plus cyclophosphamide at day 8 post-inoculation, there is suggestion here of the virus crystal in the endothelial cell. It's not so obvious as in the other one because one can't see the crystalline array. It may be possible by other means to show it better. Overall, there's loss of organelles. Overall, there's an increase in the free ribosomes. And again, there's these elongations which are associated with the pathogenesis of this disease. It's not too difficult to visualize that if the virus becomes attached to these, that this particular cell, the capillary endothelial cell, can act like a fixed phagocyte and envelop those virus particles and take them into the cytoplasm where the protein is uncoated and the RNA of the virus adapts to the DNA and RNA of the cell itself. The virus replicates within that cell. Is the typical finding that we have with cells that are invaded in these small cysts. These vesicles are present to various degrees on the luminal and abluminal surface. As we can see here, there's a considerable increase in the vesicles at the abluminal surface. These vesicles are associated with the transport systems, particularly closely associated with the Golgi system. And here one can see a mitochondria which is degenerating. Figure 94 again is the day 8 post-inoculation virus and cyclophosphamide. One can see the blood capillary endothelial cell showing large and small uncoated and coated vesicles. Here at 1 are the large, the small uncoated at 2. There are a large number of free dense particles which on size basis are free ribosomes. Here at B, there is a degenerated neurone, perikaryon, showing general depletion of organelles and increased numbers of free ribosomes and polysomes. This is figure 95, still on day 8 post-inoculation with virus plus cyclophosphamide. Here again one can see in a pericyte which not only is lysed, completely degenerated, showing liquefactive necrosis, except for a few small vesicles in the area. It's not only showing this, but it's showing the characteristic virus crystalline array associated with the presence, which is the presence, of porcine teshovirus 1 virus. And so this again is pathognomonic of the lesion. So far we have seen a pathognomonic lesion in the blood capillary endothelial cells associated with liquefactive necrosis and swelling, and now we see it in the pericite of the associated cell to the blood capillary. Presumably, this virus has got there by the dysfunctional transport system of the Golgi and been carried into this contiguous area of the blood capillary. In A, small and large vesicles associated with dysfunctional transport system in the Golgi apparatus, in which the Golgi are related to the this transport system under normal circumstances with smooth endoplasmic reticulum, and this possibly is an enlarged smooth endoplasmic reticulum associated with dysfunctional Golgi system transport. And here one can see the possibility, you see the way the cytoplasmic membrane is folding around itself, producing a vesicle, and therefore it would not be surprising to see that a virus or virus particles could be enclosed within there, entrapped, and make their way into the cytoplasm, carried by this type of vesicle. So here a large number of vesicles at the luminal and also particularly the abluminal surface. And this illustrates the disease with respect to neuropathogenesis involves a sporadic invasion, because here is a neuron that looks quite normal next to one that also looks quite normal. This is the axon hillock here. The blood capillaries here are reasonably normal. So this is a multifocal degenerative condition of blood capillary endothelial cells. Obviously some are normal, some are not. Here the mitochondria are normal in size, in numbers and distribution. The Nissl substance is intact. It's not freed up like the other ones where there were free ribosomes. And so one can see normal neurons associated with this particular disease. Here in figure 97, there are normal amounts of Nissl substance here, but here there's a suggestion of increased fine particulate matter. Some of this is less electron dense and therefore could be free glycogen as a result of the dysfunctional transport system in the Golgi apparatus, which is beginning. By and large, the Nissl substance is intact. There are, of course, here some free particles which are denser, and that indicates early degenerative change associated with this Nissl substance. You can see that the appearance is by and large the same size and same density as in the Nissl substance. So that indicates an early degenerative change. But the mitochondria in general, the cristae are not depleted, and in general one cannot say that at this stage that there is certain degeneration. And now we turn to the axon and the Schwann cell particular disease in the dorsal root ganglion. Once again, this is a day 8 post-inoculation, virus and cyclophosphamide. Here is in A is the axon showing mild increase in particulate matter, and the particles are probably free ribosomes. There are coated vesicles, odd coated vesicles here. Now here in B is evidence of an almost completely degenerated Schwann cell. That can be related to the myelinophages that we saw before and the myelin degeneration, because here we can see specific degeneration and a liquefactive type necrosis of the Schwann cell. There are a couple of rather normal mitochondria, but here around the surface one can see both inside the cell and just outside it a large number of vesicles. Now, whether there is direct attack of the virus upon the Schwann cell is uncertain here. One suspects that it would be difficult for a virus to invade this cell because of the barriers that exist and because it's not closely associated with the blood capillaries. So therefore, the conclusion is made that this is an effect of hypoxia associated with the blood capillary endothelial cell necrosis. But the question still might remain that maybe virus could attack that, but at the moment there's no conclusive evidence, nor is there logical deduction based upon anatomy to show that there's a direct attack of the virus on Schwann cells. We now turn to figures 99 and 100. A virus-only treated pig on day 8 post-inoculation. Here in A, the neurone is showing diffuse chromatolysis and vacuolation, which one can easily see on the histopathology. We saw it before. Note degenerating neurone showing widespread vacuolation or vesiculation and loss of Nissl substance. The Nissl substance which we see here in B is not present here in A. The one here in B is a normal neurone. Once again illustrating multifocal nature of this disease associated with the presence or absence of blood capillary endothelial cell necrosis of the parent blood capillary endothelial cells. So here one can see widespread vacuolation and vesiculation and loss of Nissl substance, and at least some of the vacuoles might be enlarged cisternae of smooth or rough endoplasmic reticulum. There is also a suggestion of increased ribosomal material, and higher power will reveal severe effects on mitochondria. This is at a magnification of 8,000. Turning to figure 100, which is part of the neurone illustrated in figure 99, there are amorphous areas of fine, less dense material here, which indicates free glycogen, and the Nissl substance is missing, and there is a distribution of free ribosomes from that missile substance throughout the body. Their mitochondria are mostly normal, the cristae are normal, but here there are in 3 and in 4 there are vesicles which are single coated in 3 and double membrane coated in 4, and this particular one indicates a degenerated mitochondrion associated with peroxidation and oxidative stress due to the elaboration of free radicals because of the dysfunction of the mitochondria. This is figures 101 and 102. This is day 11 post-inoculation, virus-only treated pig. Shows a neurone, the perikaryon of the neurone, showing a clear zone around the periphery here and vacuolation here. The zone here appears to be of proteinaceous material, not unlike that seen outside the cell. So here we're seeing damage to the blood capillary and the gaps between the blood capillary endothelial cells, which are normally very closely attached. But with the severe liquefactive necrosis that one has seen, it's not unreasonable that one should find evidence of progressive invasion of these areas with proteinaceous material from the bloodstream. It's not severe enough to produce a hemorrhage, but one can also see the presence of a lymphoid cell in this region, which also indicates that there might be some gap or break between the gaps in the blood capillary endothelial cells, which are normally fairly strong, particularly in the brain, but sometimes there are gaps in the dorsal root ganglion under normal circumstances. So these vacuoles are part of the vesicles of the dysfunctional transport system, but also on higher power we'll see that they are degenerated mitochondria, again reflecting hypoxia because of the degenerated blood capillary endothelial cells of the parent cells that normally would supply the oxygen to them. So turning to this final figure 102, one can see here at 1 is a severely degenerated satellite cell, which indicates that the virus has attacked directly the satellite cell without directly attacking the perikaryon. Embryologically, these perikaryocyte and the satellite cells are more closely related in their histogenesis to blood capillary endothelial cells, whereas the perikaryon is more associated with ectoderm. It could be that based upon histogenic genetic and histological biochemistry that it is less likely for this perikaryon to be affected. After all, this perikaryon does not show all the villus type of reaction that one sees with blood capillary. And so the mechanism for actually taking in the virus may not be there. And to support this, one can see along this membrane that there's no large coated and uncoated vesicles that we saw with the blood capillary endothelial cells and in the perikaryocytes. But what we see here in number 3, membranes and remnants of Christi. So this is a severe peroxidation and oxidative stress of the mitochondria associated with large numbers of blood capillary cells being affected. Using the Watt theory of relativity, mortality and morbidity rates of the dependent cells, like these here, is directly related to the number of degenerated cells represented by the mortality rate of the blood capillary endothelial cells. And so this varies largely depending upon the immune status of the particular pig, the particular animal, or the particular man at the time. As we can see, with the virus plus cyclophosphamide, the cell-mediated and then the humoral antibody response is lost or severely restricted. And so that means that a very large, much larger number of blood capillary endothelial cells are permitted to spread by systematic contiguous invasion, and so that produces the more severe clinical signs and the greater distribution of lesions than one would see in the more moderate form, which is restricted by the presence of the cell-mediated response and later the antibody response from the B cells and then the plasma cells which produce the antibody. And now we come to the summary and conclusions arising from this epic journey of discovery. Now, the Watt neurogenic bladder could be the basis for high mortality seen in the 1930s with the so-called highly virulent Teschen strain, and in Haiti in 2012. The reason for this is that the neurogenic bladder is actually a result of domination of the sympathetic nervous system over the parasympathetic nervous system and the closure of the sphincter of the neck with continued urination. The bladder gets bigger and bigger, and so the sympathetic nervous system comes into play and causes increase in blood pressure and cardiac rate, which can be fatal. Obviously, in a natural outbreak in pigs, that would not be attended to by the doctors in the case of of humans, in the hospitals they would catheterise the bladder. This occurs in conditions like traumatic injury, in accidents, and the localisation of the lesion is from T thoracic 6 upwards. And so when you get these lesions from, say, spinal cord trauma or even poliomyelitis, infections at T6 upwards, you get dilation of the bladder. And this can then lead to fatal cardiac failure. So we have this rather interesting situation where the pigs, instead of dying from neurogenic failure, in the brain and spinal cord, they died from cardiac failure. The second original finding, the unique original isolation of porcine tissue virus 1 in Australia from a point source epidemic. It was unique because we isolated it in 1970 and twice over the next 6 months, and it's never been isolated since, 44 years later. And so the reasons for that could be because of the fact that the pigs were running on a deep litter system, whereas that is not the sort of system that occurs these days. They run on boards or on concrete, and the feces is not present in large amounts like it is in a deep litter system. So it's a matter of dosage as well. And it's interesting that in Europe, when you visit a farm, quite often the deep litter system is present next door to the bedroom and the lounge room. And so they have had deep litter systems in the past, and maybe they're still there now. So it's a matter of dose rate and the presence of fecal feces contaminated with the virus. From this isolation arose a dilemma. And that dilemma is like an oxymoron. How you can have something that's exotic and endemic at the same time? Exotic means it's outside Australia, endemic means it's present throughout Australia. And that's why the Teshin Talfin is the cause of a dilemma and a situation which does not really exist. And I proved that by using immunosuppressives. Using the mild form, I created the massive, highly virulent form, so-called Teshin. So this is evidence that the so-called two-strain virulence does not exist. And that's also supported by molecular biology, in which there's no evidence at all that there's any molecular biology variation between them. It's a common serotype. We now turn to the next original discovery, and that's the white cell nodule or glial nodule. For 80 years or more, the glial nodule or cell nodule has been recorded. It's involved in a great range of viruses attacking the nervous system. Previous authors have recorded it, but never explained why it's there. The white cell nodule explains why it is there. It is there because of a primary initial attack of the virus on the local blood capillary endothelial cells. There is a reaction of the tissue to the presence of that necrosis. The cells present are a mixture of glial cells and of hematogenous cells, lymphocytes. So it's not really a glial nodule as such. It's a primary oncotic necrosis, focal necrosis of the blood capillary cells. That is the initiating lesion in the neuropathogenesis of porcine tischovirus disease, and also of a number of other viruses affecting man and animals, which I'll deal with later. For example, human polio is currently considered to be attacked. In human polio, the virus is considered to attack the neuron directly. This contrasts with evidence produced in the 1960s by Blissinger that the virus is present in the same places as I found with my piece of research. So the same pathogenesis is likely to exist. And that is, when these blood capillary cells are destroyed, there is dysfunctional transport of nutrients. The normal food, gas, and water that goes to the dependent cells is rendered dysfunctional. And that's reflected in the appearance of the pathology on light microscope studies and on electron microscope studies. When the cell nodule was examined under histopathology, one actually used sagittal sections which revealed the lumens of the blood capillary cells. Now, when I did that doctoral study, I wrote that this was an increase in numbers of capillaries. On reflection, and recently, from the work of McGovern and Robbins in the basic textbooks of pathology, I've extrapolated from wound healing, in which they state the wound produces hypoxia, and those blood capillaries are reacting to hypoxia. So this reinforces my research in that the hypoxia is produced by destruction of those local blood capillary endothelial cells in the brain, the spinal cord, and the dorsal root ganglia and the trigeminal ganglion. And so the neuron, which one could call, I've called it the what neuron because I want to draw attention to the fact that it's not attacked directly by a virus, that it's a secondary effect caused by primary destruction of the parent cells, with secondary effects upon not only the neurons but the other cells in the nervous tissue. So with this neuron, which one sees under light and electron microscope, one can then read the slide now, which one couldn't do before, and that is the presence of free ribosomes, which are been lost from their normal place in initial substance. All these molecules they're held together by energy forces, and if the energy is depleted, as it is with this lack of nutrients, then this results in the loosening and freedom of the ribosomes. So one sees diffuse ribosomes, and that is explained by lack of energy. One also sees in the neuron, with the what neuron, vacuoles of various types. The explanation for this is the dysfunctional transport system, particularly in the Golgi apparatus. And so one sees marked enlargement of the smooth endoplasmic reticulum and a whole range of other vacuoles. And this is principally water. It's either water or gas. And it's most probably water. And therefore, that condition is called cytotoxic edema because it's within the cell. That contrasts with the blood capillary cell, which shows oncotic necrosis. It's also that blood capillary is also filled with water, and it's there in that blood capillary there is complete lysis. One usually does not see complete lysis of a neuron. It's a gradual process and it's a varied process, which indicates lack of oxygen and other nutrients. In contrast to a blood capillary cell, which is invaded by the virus, destroyed, where the virus multiplies and destroys the whole contents of the cytoplasm, and this disrupts the sodium-potassium pump. Larger amounts of sodium ions are present inside, and so water comes in and the cell swells, called oncocytic. A critical lesion inside the cell is the effects on the mitochondria, the degeneration of mitochondria at various levels of severity. The mitochondria may show hypertrophy, hyperplasia, and also destruction, degeneration of the cristae, associated with the generation of free radicals by an aberrant cytochrome system. And so, with depleted, degenerated mitochondria under anaerobic conditions, there is marked depletion of energy which affects all the dependent cells. The next discovery was the microinfarct. This was associated with coagulation necrosis, and coagulation necrosis is caused by hypoxia, which once again reinforces the evidence of blood capillary destruction of the parent cells. The next contribution to science is the brain topographic mapping, in which there is a qualitative and quantitative mapping of the whole brain using whole brain transverse or coronal sections. This shows the distribution of the lesions throughout the whole brain, and also it shows whether the lesions are bilateral or not. And if they are bilateral, this indicates that the origin of the virus is from the bloodstream, as we did discover. Now, the next evolvement was the so-called Watt theory of microcirculation pathology. Here it was demonstrated and also theorized that the virus is attached to the blood capillary cells because of a slowing down in the blood flow in the microcirculation at those particular areas which have high energy needs in order to sustain life, basics of life. And so these lesions were found in those areas, in the cranial nuclei, the spinal cord ganglia, and in the dorsal root ganglia. They're very large neurons and they need a lot of energy. And so it has been demonstrated that there's a higher complexity of blood capillaries in those areas. If you increase the blood capillary complexity, you increase their surface area. Therefore, you increase the frictional forces. And so this, combined with the morphology of both the blood capillary endothelial cell that we saw ultrastructurally and the leukocytes and even the thrombocytes that we saw ultrastructurally, combined to slow down the blood supply. By this I mean, on the morphology of the leukocytes was that they sent out so-called pseudovilli or pseudopodia, and those villi obviously increase the surface area. And the same occurred in the blood capillary endothelial cells. And so the combination of the complexity of the blood capillary cells and those villi forms in both the blood capillary cells and in the leukocytes and even the thrombocytes had a slowing effect, and therefore by random chance the virus particles got off their ride on the leukocytes and became attached to the blood capillary cells, subsequently invading them by a system of caveoli and then destroying the cell. We now come to what happened to Harold. You will recall that Harold was seen with severe posterior paresis, and Harold, at 23 days after the first sighting, was improving. On day 44, Harold was completely recovered. This has great implications for human patients. To try to explain what was going on at the cellular and subcellular levels. The first thing is to look at the lesions that were present, and it hasn't been acknowledged or recognised in the past about the severity of the lesions in the white matter. And so the lesions in the white matter obviously produce demyelination, which slow up the nerve impulses. The body, it has been shown that the body has the potential to remyelinate through the oligodendroglial cells, and so remyelination can occur over time. The other thing is the a withdrawal of possible of water from within the cell, from an edema, cytotoxic edema. In milder forms, there will be regression of that water. The other very important thing is the possibility of hypertrophy and hyperplasia of the mitochondria, restoring the energy, because the mitochondria have their own DNA, and by epigenetic methods can actually reproduce. And so the mitochondria could be restored, the water restored, the myelin restored, and finally is the lesion that we saw on histopathology in natural cases of neovascularization caused by hypoxia, just in wound healing, same as in wound healing. And so one can see these new endothelial cells, which are larger than normal because they haven't got the blood in them at the moment. When they get the blood in them, the pressure of the blood will flatten those capillary endothelial cells. And with the regeneration of those blood capillary cells goes the restoration of blood supply. And so all these things that I've just talked about to you, restoration of blood supply, remyelination, withdrawal of water and restoration of the mitochondria can explain how Harold recovered.